Welcome back, everyone, for the afternoon session. Very excited to have a panel discussion here. So I think through the conference, there's been a lot of really interesting talks, but questions and points that have been brought up as well. So I think this is a great opportunity to go through some of those, have a very open discussion. So we've got the panel answering questions, but I think the intent is to make it such that anyone in the audience can obviously ask questions, but also contribute in the answers as well. So we'll start it off with each of the panel members just quickly introducing themselves, who they are, and outlining some of the challenges that they see in the fields and top predictions for where the field is going over the next few years. Then I'll ask a few questions, and then we'll open it up to the audience. So Paul, start with you, please.
Well, good afternoon, everyone. I wasn't expecting to be here, but thank you very much. So I'm a professor of analytical science at Loughborough University, and I've been interested in breath analysis since about 2000. And I've been interested in volatile organic compound analysis since 1984. The two challenges I'd like to put out there, this is such an advantage, by the way. The two challenges I'd like to put out there. Challenge number one, what are we dropping? There are some really important compounds in breath, in our systems, that we don't report and never get cited as potential markers. And I know they're there, and I know they're important, and we're not seeing them. So I think challenge number one is to pick up the molecules that we're dropping and to start reporting on those. And challenge number two is, before I die, I'd like to see an agreed, normal, standard human model breath profile. What constitutes healthy breath? What's the variability? What's the composition? There's the base composition of human breath, and then what else is attributable to everything else? And that, for me, is the scientific interest for me. Thank you.
And what do you think over the next few years? What are your kind of big predictions for the field? 5 years ahead, we have our 7th conference. What is the kind of key thing that will have happened by then?
5 years from now, our prediction number one is we will see breath analysis being used on specialized, highly functional clinical processes. And I'm thinking anesthesia in the first instance, and maybe some toxicology screening in the second instance, but very specific, highly controlled clinical interventions 5 years from now. I think we'll start to see the first sets of theranostic patents being developed and going out to wide-scale trials by then as well.
Excellent. Mark.
Great. Hi, I'm Mark Vandershay. I'm the head of clinical science for Owlstone. Been involved in breath research for not as long as Paul, not nearly, but for quite a while, almost 13 years now. What I'd say is the thing we really need to focus on for the next few years is fundamentally starting to understand the biology. And with that, I mean the biology of the production of VOCs by understanding, by performing very cleverly designed mechanistic experiments, some of which we've seen examples of during this conference. But another aspect of understanding the biology is also the mechanics of how these VOCs ultimately end up from being produced in the cell into the breath. How does the physiology of a patient impact, and how do you collect that ultimately in the most reliable and reproducible way? And probably we're talking the most reliable and reproducible ways, because this could differ depending on the applications and the constraints. And in terms of where I see ourselves in 5 years from now, I do agree that I think that kind of fundamental biological understanding will first enable you to, in specific applications, sort of launch these tests where perhaps the sort of addressable clinical problem is not as big as sort of one of these major tests, such as early detection of cancer. But you can very clearly, through the mechanism, demonstrate that there's value in a breath test. I think once you get to that point, that proof of concept almost, that's what starts driving the necessary research and investment and collaboration into larger scale trials. And my ambition and expectation is that in 5 years' times we will be quite a ways through these kind of very big validation studies where we're looking at tens of thousands of patients for these massive applications such as early detection of cancer.
Excellent.
Thank you. So, Renaud Louis, I am Professor of Respiratory Medicine at Liège University. As I said, I've been involved in clinical research in asthma for 30 years. I have only a recent interest in breath analysis because we liaise with my friend here, first in Maastricht, and then we reproduced that with the chemistry department in Liège. So what I understood from this morning session is that really we need, I think, huge database to be confident with the markers that you could validate. So I think for me that's the challenge one, is to try to be able to set up some consortium that will validate biomarkers, and because of the great influence of environment on the VOX that you can measure in breath, I think you have to probably to be widespread in your consortium, because I'm sure that the environment here is not the same as in West Germany or in south of Europe. So I really believe it's a major issue for me from what I heard today. The second challenge is more something which deals with the financial aspect of things. I'm really struck by the fact that there is growing progress in biotechnology. I'm fascinated by the work of chemists, engineer, chemistry, and I think there is a growing gap between what we can do on a technical point of view and what we do in clinical practice. And this is something which is due, obviously, to the way we finance the healthcare system. And I think it's probably a great challenge for the 5 to 10 years in the future.
Okay, I'm Frederick Jan van Schooten. I'm professor of genetic toxicology at University of Maastricht in the Netherlands. So I'm coming from the field of environment and chemicals and how they are inhaled and damage DNA and how it influences DNA repair. So it was mainly on lung cancer. And actually I entered the field around 2000 when Michael Phillips published a paper on lung cancer and using exhaled air. So I was fascinated by that. So originally I was working on lung cancer, and swiftly we went to more internal organs as the colon and the liver. So that's how I entered this field. The challenges, what we should do is understand the biology, where these compounds are coming from. If you're talking about endogenously formed compounds, if there's information in it anyway, but we should know which biochemical pathways they are coming from. So for that we need good in vitro systems, cellular systems or bacterial systems, but also experimental animal systems. So I think that's a challenge, to back up all the information we can get and also to convince the physicians that it's really something they are looking at. And of course what we should do is to have good external validation studies, clinical validation studies. That's the 2 points for the future. Concerning to me.
Okay, good afternoon everyone. My name is Jonathan Beecham. I'm a physicist with a PhD in environmental chemistry, and I'm working at the Fraunhofer Institute in Freising, which is in South Germany. And I've been working in breath for about 15 years now. During my PhD I started. And Paul alluded to it was the easiest, the question was easiest to answer at the beginning, so some of the points have been raised already. I agree with Mark that understanding the biochemistry of these compounds is critical to understand what's going on in the body and to actually validate these biomarkers. In terms of what's going to happen in the next 5 years, probably not Brexit, but in terms of breath, I think that the challenge is most of the breath research at the moment is done on a lab basis. So these are big analytical instruments. You need a lot of training to actually operate them. They need to be calibrated. And I think miniaturizing these tools or having sensor systems that can actually do a similar job to these tools that can be used in the point of care and be operated by laypersons just that have a small, brief introduction to the technique. I think this is a challenge. I don't know if that will happen actually in the next 5 years, but I think this is the future of breath if it's going to actually make it into the clinical setting.
Okay, it's my turn. I don't know. Maybe I am working on artificial intelligence for many time. I prefer don't say the time because I remember my age and it's not so. But I teach in the chemical engineering department, and as always, I always say to my students about the money. What about the money? It's important to have. I think one challenge will be propose equipment, a powerful equipment, but with a price that many people can allow. Test his breath in this equipment. This is I think for me is the most important challenge because we need to put everything that we do in the street, in the patient, in the clients. I think this is very important. And also the challenge in these 5 years, I think, will be remove all type of wall between disciplines. One chemical engineering could be work with other discipline, doctor or whatever. I think this is the secret. I think that this is the key to in my opinion, this is the key to find many solutions. And also the consortium. I have the possibility to the third challenge is don't have any problem to in a mathematics, for instance, project focus on the breath analysis. Doesn't matter if you propose a psychiatrics or a psychologist department. I don't know. We are going to open the group the group that can form this consortium. But my main important challenge is reduce the price of our equipment. It's very necessary to put this equipment to the patient, and every patient can have this information for everything. Thank you very much.
Excellent. Okay, thank you, panel. So we'll start with a couple of questions that were submitted before. So I think one of the themes from yesterday and today, when you reflect back on the history of the fields, there's actually very few breath tests which are out there in use. So, some of the stable isotope tests, exhaled nitric oxide. So the absolute pre-requirement for any test is the biomarker. Without a biomarker, you're snookered, basically. So we've also talked a lot about the different potential applications. So, in asthma, in liver disease, in cancer exposure. So I just want to get a sense from you, and it's been touched on a little bit already, but also from the wider audience, where do you think the kind of next advance is going to be in terms of finding and validating a biomarker in a clinical application? So grab the microphone, whoever wants to start, and then we'll also open this up to the audience as well if you have a view on this.
So if I may start. So we've seen a lot of the talks yesterday and today, or several of the talks, they showed data from the so-called EVOCs and exogenous VOCs. And this is the catalyst was Chris Mayhew's paper that showed the elevated limonene levels in patients with liver impairment. And I think this is really an exciting application, and I think that's reflected in these talks. Everyone's talking about it, that it actually has huge potential. And I think it's great that Owlstone is dedicating some time on that, looking at these EVOC probes, because I think this has high potential to be a clinical test for looking at the functionality of certain organs, especially with the liver. And seems a lot more promising than finding the elusive lung cancer biomarker that no one's found in 15 years, as Anil was already talked about yesterday. Mm-hmm. So that would be my expectation, that this has a lot of potential.
I just want to add something on that. As Paul said, don't throw too quickly away compounds. And for instance, we did this liver study and we found limonene, and we were discussing in our group, it's an exogenous compound and we should discard it from our analysis, just throw the compound away because it can't be endogenously be formed. So, but then our discriminating model dropped down. So in the end we included it, and in the discussion we tried to explain it at that time. So there was hardly we should have thrown it away, because and at the time I think we were quite harsh on each other when there were some, let's say, compounds in it that you could not explain because they were coming from environmental pollution or whatever. That it can't be any biomarker, of course. So, but now this limonene, let's say, story makes me quite enthusiastic that we really could use maybe these probes also when you are exposed to it by food or whatever, and how you handle it, the body handles it, and you exhale it. Maybe there's information on a liver, for instance. So I'm quite thrilling about this idea.
So that's 2 votes for liver so far. It's like, what's our favorite organ here, basically? Two votes for liver.
If I'm entitled to 2 votes, just to make this, oh, you're being very strict here. I'd say, I think given what we've seen, the evidence for limonene is quite compelling, and for EVOC probes in general here. Just a slightly challenge on the other thing you mentioned around the elusive biomarker for lung cancer. Yes, it has been elusive for a long time. At the same time, what really excites me is now we're getting to that concept of an EVOC probe and understanding the biology better, that what we've seen at Owlstone is actually getting people on board that really understand this biology in detail. And there is, I don't think, any other disease than cancer that has its metabolism and biology as well studied. So going through that and trying to find specific targeted strategies to ultimately identify what patients suffer from cancer, yes or no, or are at increased risk of cancer, I think we'll also see very significant progress there over the next few years. So that's sort of a split vote.
Can I respond on lung cancer? So there are many imaging technologies to see quite small nodules or starting tumors in the lungs. So I wonder, when you have this small nodule or whatever producing maybe some specific chemicals for cancer, how the F can be a detection system for early lung cancer. So I question that. So maybe someone disagree with me.
So I didn't want to insinuate that we'll never find any biomarkers for lung cancer or any other cancers. But I think it's been a challenge. I think one of the challenges has been that people have maybe not taken as much care or weren't aware of what they need to be aware of in doing these clinical studies. There's certainly a lot more awareness nowadays on standardization, cross-validation, also in terms of data processing. We have a lot of improvements in mass spectrometry, high resolution, gas chromatography, mass spectrometry, where you're finding a lot more compounds and you're more likely to find something that you may not have found before, also at the levels, the trace levels that perhaps previously couldn't be detected. So I think there is still a future in finding these biomarkers, but the more immediate future is doing something like with the limonene. That's why I mentioned it.
I would like to stress again the importance, I believe, to take into account environment, and given the high variation in quality of hair within Europe, for instance, as I said, West Germany is not Scandinavia, it's no south of Europe. I really believe we need, if we want to identify the VOCs which are really linked to the disease process, we will need, I think, large-scale study conducted in different parts of Europe and taking into account the different variation in quality of hair.
Could I just ask a quick question? Trying to think about where we'll see the biomarkers in applications. So I think the work you presented earlier on the VOCs and asthma phenotyping is really very impressive work that we've seen. Do you think that will translate ultimately to validated biomarkers?
If this is confirmed, that was our intention, obviously, because if this is confirmed by other groups, but I think it needs to be confirmed by other groups in the world, in Europe. But if yes, I would believe it could have an application in clinics, for sure, because it's much easier to detect some VOX compound in exhaled hair than to do sputum induction and process the sputum. It takes 1 day, just 1 minute. So, and I think it's key to drive the treatment. And as I said to one of your colleagues, it will cost money, but you could have as an argument that you could also spare money linked to, I would say, inappropriate prescription of drugs in patients who are not responding. So I think that's...
Well, 3 points, if I may. I'd like to pick on the we're being harsh to ourselves. So Arthur C. Clarke said you don't know something's not possible until you've tried it and failed. And at the boundary of what we're doing, we're always stepping into things that are currently not possible. And we know so little about what it is we're trying to do and the processes we're studying. And we're very quick to be harsh and judgmental, particularly when people are trying to report on observations. And our ignorance is still profound. So I'd like to endorse that point, if I may. In terms about cost, I really want to emphasize the importance of enabling data to be shared. So without wishing to sound boastful, I've got over 120,000 curated breath data files on a server at Loughborough University, and I am aching to share them with people. I want people to say, I want to do some validation. I need as many breath samples as I possibly can collected in a reliable way that I can test my findings against. And it's through that collaboration and coordination that you will address the second point, what about the environment? So you have geographically resolved repositories of data collected locally and shared. You get massive leverage of the international research effort, and this is really important. It comes back to the standardization issue that Jonathan was making. And then finally on the environment, I believe that breath research is going to drive the most comprehensive and wide-ranging characterization of indoor air in hospitals that we have yet seen. We're currently as a result of work with colleagues in Leicester, looking at 2 years of continuous trace VOC monitoring of the clinical environment. And what we will know very shortly is a probabilistic model of clinical VOC profiles. And similarly, other breath studies are doing exactly the same thing. And just looking at that data in and of itself, I think produces great value to the whole communal effort. So this is what we're doing, I think.
What's your favorite organ?
My favorite organ?
What's your favorite organ? Where is the biomarker? Where's the biomarker going to come? Lung, liver, tumors? We'll get T-shirts made for next year. We can all wear.
All right, I'm going to be mischievous, if I may. So my favorite organ is a sexist organ, and that would be the brain. I think the key between psychological state, emotional arousal, and biochemistry is going to be really important for us. And so my favorite organ is going to be the interaction of the brain with the VOC production mechanism.
Excellent. Anything before we open it up? Yeah, we haven't had a response from Jose.
My favorite organ is lung cancer. Lung.
Yeah.
And also I agree with my partner, and it's the connection between everything with the brain is extremely. I love this type of things. Between body and the brain is one of the things that I would like to study more in detail. And depending, and also about the biomarkers, I am going to I agree with the study of profiles. Profiles for me is more useful than biomarkers, because we have a problem. We have a real problem. This is the biomarkers of the profile is different depending on the region, depending on the continent. But in real, if we have enough databases, we can put all together to work in maybe not in the same model, but we can work in different type of models, but we can propose a solution, a common solution in a more complex model, but using this type of profile better than biomarkers only.
Okay. Good, so we'll open it up to the audience as well. So it's the same question, where you're thinking VOCs are likely to yield biomarkers. Should we start with Bruce here?
Okay, well, I'm not a breath expert, but my background is in oncology and genetics. But just a comment on the lung-liver discussion. It seems to me that most of the effort at the moment in the liver is not so much on diagnosing hepatocellular carcinoma, but on assessing liver function. And the point there is, I think it's much more likely to succeed because you're looking at the function of a whole organ. When you're trying to look for a cancer, you're looking for something different in a small number of cells, because you're interested in early diagnosis, which is different from all of the cells that are on the way to being cancer across the whole tissue. And so coming to lung, I have particular interest in the injury to the airway epithelium caused by smoke exposure, and identifying people who, because of their background, respond differently to that and who would be at higher risk. And so I don't rule out that we will find a marker for lung cancer itself, but by analogy with the liver, it seems to me that looking at the oxidative stress response in the epithelium in response to smoke injury, something which we already know a lot about, in fact, and the genes and the regulatory mechanisms are involved. And it may be that there are different individuals who are current smokers who have different degree of oxidative stress response, which makes them more likely to get cancer. And particularly in people who've given up smoking, who account for more than half of the lung cancer burden, and they are motivated people to take part in studies because they've already been motivated enough to stop smoking. The question is, are some of those more likely to get cancer than others? And the likely biological mechanism, for which there is some evidence, is a persistent inflammation. So I would put my money on looking at those changes in the tissue and looking at risk by analogy with the sorts of things you've been talking about in terms of fatty liver.
Fantastic. Oh. Okay, go.
I'm really curious about the subset of gases that are shared by all humans. And the only paper I remember that looked at that was one by Dr. Phillips a long time ago, when he estimated that there were probably less than 25 major features shared by all humans. And since Owlstone has now got so much data from humans, I'm wondering if you've looked at that and how many major features do we share? That would help us then begin to tease out what is not in that shared subset that's due to disease or exposure.
We'll come back to that. So we'll bring questions up into the panel. So I thought what we're trying to do is go through these first two questions first. And then, so if you have a view on where we're most likely to see the next biomarker coming out.
I think it's in brain, and I think it's in psychiatry, and I think it's in schizophrenia. There are excellently validated studies now by many groups finding a cluster of gases, a profile of gases that are distinguishing schizophrenics, and it's not yet being used for diagnosis or to monitor treatment, but that would be a real breakthrough.
Have any of them been externally validated, though?
Yes. If you mean different groups redoing the same methods and getting the same results, yes.
Okay. Mark?
Just to continue on the point that Bruce made, which I think is quite crucial, and about a year ago, I think I was asked to join a ERS meeting, the European Respiratory Society, and there's a lot of evidence, and it was raised by Fredrik Jan as well. There's imaging, and this can show small nodules, and this is continuously improving. However, something I hadn't quite realized, and was a bit of an eye-opener, is they said, well, the ruling criteria for CT screening, they depend on age and smoking history. And age is easy, but smoking history really isn't. It's actually very difficult to look at your population as a government and say, oh, I'm going to send these people a letter because they smoke this much, because this information is captured nowhere, and is actually compounded by an increasing problem where the environment, which was also raised here, is becoming a more and more important driving factor for the development of lung cancer. So if you combine the two, the current sort of risk criteria are expected to drop, and currently less than half of the lung cancers are captured by these high risk criteria, and it's getting worse and worse. So perhaps it's a matter of reframing it, and like Bruce was saying, it's not about diagnosing this small tumor, but it's about finding the population that has this continuous response in the body to exposures that predisposes them to disease. And from my perspective, this could be anything ranging from lung cancer, the obvious one, but also exacerbations in COPD and asthma, which is profoundly associated with environmental exposure. And we've seen from our own data that actually exposure is something you can very readily pick up in the breath. So that's at least sort of, from my perspective, how my thinking has shifted in this specific space.
Good. Okay, so question number 2 that had been submitted, and it was touched on a little bit earlier and alluded to a little bit in the panel. So there's almost 2 different philosophies, when we try and attack the problem. One is more understand underpinning biology, make sure that we're doing chemical analysis. We have the ability to ID those chemicals and link it back to the biology, so a mechanistic-based approach. Versus the more top-down data-driven approach where you're more reliant on collect a lot of samples, look across all the features within that, and rely on statistical models and validation of those models with new prospective cohorts. So I just wanted to get a sense from people on the panel and the wider audience, where would you put your money? Where should we focus? You have one chip to put on the table, where do we put it?
What I can say from a pharmacological perspective in my field, in asthma, is that it's more the bottom-up strategy that pays, because the most recent progress were identifying a compound which was selected as pertinent based on physiological study and then test that in clinics. But today we don't have any drugs that comes from a top-down strategy so far. But
anyone who's worked with me or has been taught by me will know that I run essentially on 2 rules. The first rule is assume nothing, and the second rule is trust no one. Given that every time something goes wrong in my lab, I can trace it back to breaking one of those 2 rules somewhere along the line. I would say if you don't know what it is you're measuring, if you can't name it and prove that that's what you're measuring to a high standard of verification and validation, then, you may as well take another magic stick or special feather and use that as well. It has to be tied down. The molecular identities have to be tied down. You can then create a sensor that monitors those molecular identities in a robust, reproducible, controlled, and verifiable manner. but you can't do discovery, and you can't do difficult monitoring at low concentrations in uncontrolled environments with that. I'm going to stop ranting in a moment, I promise. My final observation for us is if we want to look at the best funded, best equipped, most experienced teams for trace gas analysis on the planet, who are not resource limited by any sense as far as we're concerned, they have infinite resource. They use molecular identity as the basis of their detection systems. And at the very least, they'll need a mobility under controlled conditions to raise an alarm on, but they don't use a statistical driven approach. They use an approach based on the detection of known characteristics of whatever poison is they're trying to detect on the battlefield. I'll stop now.
Mm-hmm. So tell me, who are these people?
the U.S. military. Yes.
Exactly. Very simply, bottom up and biology driven. Fundamentally, just to add to what has been said here already, to my knowledge, there's also no diagnostic currently approved that is purely based on pattern recognition, simply because even if you would solve all these engineering issues and would do massive and very, very expensive studies, because you would need enormous amounts of patients, you get to the point where if you try and seek regulatory approval, you will never get it, because you're unable to calibrate your device. You're unable to show that the next device does the same thing as the previous device. And for clinical diagnostics, that's simply unacceptable to a regulatory agency. So that just compounds the challenges that have been mentioned by the other panel members here.
Okay, then I will take the other side. So, so you don't trust anybody, Paul, but you trust God. So if there's a God-given sensor and you prove that it's really working, so what does it matter? If it works, it works. And you can treat patients based on the sensor, God-given. And why should not, is there a principal objection against it?
What's your definition of works? That the model holds up in an externally validated population?
And it holds up in several validation studies. For instance, if you have the dog, and the dog can sense this infection, this Clostridium infection in the hospital. So the dog is entering in the room, sniffing at the patient, and it's infected or not. So that's what's done in Amsterdam at the Free University. So you do not know what the dog is smelling, but yeah, this God-given nose is working. So, and...
How would you determine it also works if the dog has a cold?
Studies have been done with lots of dogs, and they can train any dog to do it if they start them young enough.
Yeah. So how would you prove time and time again that each dog can do it? And like you're saying, you'd have to train each dog. Is that going to be economically viable?
Many dogs, and they had validated cancer patients versus non-cancer to test them against, and their sensitivity and specificity is better than the doctor.
Yeah, which to me is a testimony that there are VOCs. However, practical implications are huge. You cannot just do this in the clinic in such a way. At least I see.
There may be difference in this regard regarding diagnosis and treatment. I think I would accept that for diagnosis, maybe you are not forced to know which molecules are involved. But when you think of treatment, all the treatment in disease are targeted against some compounds. So I think if you think of Vox in predicting response to treatment, I believe that it's probably better to have a bottom-up strategy.
Okay, come back this way.
I'm just coming back to the dogs and olfactory detection. We heard about, was it Oscar the cat this morning? And also Joy Milne and the story about her detecting Parkinson's on her husband and detecting it also on other patients. And I think the work being done in Manchester at the moment is fantastic. So we don't know what this lady is smelling. but they're using GC olfactometry they've trained this lady to operate or to sniff at a GC olfactometer to actually identify the compounds that are eluting from the column to try and hone in on what compounds is she actually detecting. So I'm a proponent of understanding what chemicals you're actually detecting rather than screening for patterns.
Okay, Jose?
I'm thinking, you think, I'm sure that you think that I am going to tell you about the pattern more than, well, no, sorry. No, for me it's very important to know what is the biomarkers. It's very important to have information about the biology, but I think that once we have this information, we have to work in both stages. I think that we have to analyze everything in whole profiles, select what is the best sensor, and also continue working with the knowledge of the biomarkers, of the metabolism, many things. I think this is my opinion, and I think that this is based on the in one of my slides when I say many time ago you can diagnose viruela. Viruela is in Spanish is viruela. In English, I don't know. A type of illness that there are many grains in the face. The people who realize that this person has this illness don't wait to analyze in a biochemical way what is the performance and everything. Go for this. And this is for me, is necessary to know what is the base, and also, when I have this, start in both sides.
Okay, so I could hear rumblings in the audience, so I expect a vigorous debate here. Morad, I think, was first.
Thank you for... I'll start with you. Correct me if I'm wrong, but imaging is patterns in medicine. See, you started. I'll start with Paul. I think you said the most important thing. We still don't know enough and what a healthy population is, what a healthy breath looks like. And I think we still need years to understand that variability, biological variability, before we jump into diagnostics. I salute you for that. Bottom-up or discovery, I think if in ideal world, I would go with hypothesis-driven analysis. But do we know enough to do that? This is what I doubt. Because everyone here who published a paper in breath analysis, in his discussion, in her discussion, it goes, oh, branch alkanes is inflammation, reactive oxygen species. We don't...
That's a different thing we were talking about, I think, here.
Let him finish. Let him finish.
And what we're looking for is a unique metabolic pathway that would work in a targeted delivery, which is extremely difficult. If we go back to lung cancer, for example, I think looking at the pharmaceutical industry trying to deliver drugs directly to the cancer tissue and failing because of that lack of uniqueness. And therefore, that's the weakness of this. But I agree, if we find a pattern or signal compound and it's externally validated, it works, and I have no freaking idea where it's coming from, it works.
Okay.
So I would combine both and see what would work first.
So Chad, Olga, and then this gentleman here.
Yeah, adding a little bit to what Morad said, if we find a pattern and it works, I think that's great. And when we look at drug discovery, a lot of that is very top down. You screen 100,000 compounds and you find what works, and then a lot of work then goes into showing why it works. So, and I think something that would be critical if we do go with these pattern approaches is trying to understand what these sensors are detecting. And if that's something that we can start to learn in more detail, or for a dog's nose from a neurobiology perspective, then we can validate these things. It doesn't necessarily need to come before the sensors used in the clinic, but I think it's something that would need to come after in order to increase the confidence and allow these to spread. And that was
So is that a +1 for top-down analysis, but it's important to know the chemical ID?
I'm a non-registered voter here. So.
I'm supposed to support the validation route, identification and validation route, because I come from the assay development, optimization, and regulation side of things. However, we need to take into account that the whole route was developed and based on one target biomarker. And that's where it started, and that's where it was easy, and that's where it wasn't easy, but at least it was doable. We are moving away from this into more and more target biomarkers coming into the signature, and it's becoming more and more unsustainable, non-sustainable, less and less sustainable to go down this route. So we can imagine the situation where it is actually, and also we're building the computer machine learning power in parallel. So in the same way as we pull together all the uncertainty and variable of biological variability into higher number of samples and pushed into analysis and into machine learning, we can probably imagine, I'm not advocating that, but I can see it going this way, and not in the foreseeable future, because the regulatory won't give us this way of doing things, into actually pulling inter-instrumental variability into the same unknown variable. And then we are coming to the situation when we have not one dog, but 5 dogs, 10 dogs, 100 dogs, million dogs, and we just prove that they all do the same stuff equally well, robustly, reliably, repeatably, reproducibly, etc. So it basically doesn't feel impossible that we are actually going inevitably in this direction. So even though I shouldn't be voting this way, but probably in the far longer term future, support it.
Okay, thank you.
Thank you very much. Excuse me. Carsten Müller, Cardiff University, and I'm possibly coming from a completely different angle. I'm a trained chemist and I've spent my life as a chemical biologist, so I've been dealing with a lot of animals who quite obviously communicate using their noses, and you name me an animal without one. There are a few, but they are really rare. Now, that leads to two things. I want to find information, and it's usually in the profile. So possibly it would do everybody a favor, first trying to find a feature, which I like about pattern recognition, and then deconstruct all the noise. Because if you find 1,000 volatiles, it's highly unlikely that any organism uses them all. But we don't know which ones. Similarly, in the production side of things, I don't necessarily think that one cell different in a liver will change no, it might add something, it might distort a profile. So it's very much down to, and that's still something which sort of bugs me, and is an ongoing data evaluation process. How do I extract a pattern without jumping immediately to individual compounds? I'm a chemist, so in one way, that's the confounding thing about olfaction. It's a mixture of compounds, yeah, that's analyzable, but it is still a simultaneous impression. Going back to the body itself, it is a complete organism. It's not just a liver that has a cancer or something like it. And I think stepping back and having a little bit of an ecological look at things might really get the dichotomy away. And where my money is, in terms of medicine, I say, well, if it can be reducibly proved, it can be calibrated, I do believe that. And I don't believe it comes to the point, well, if somebody makes voodoo. But dogs do sniff out things, and the nurse does. And no one knows how olfaction works. So even if we find the compounds, we still don't really know how that triggers the brain. And I would say, and it could be that regulatory authorities will follow suit, as they did as well in the past, to say, right, there is a mixture. I love to know the molecular underpinnings, but also I think what was really important about Renault's work was to show we don't even understand the diseases. So we asked, we want a biomarker for asthma, then we discover how many different types of asthma are there. If we interrogate the same data again, we might find the modeling aspect might actually be helpful exploratively and say, I see 12 patterns. You only give me 4 diseases. So step a little bit away from this very simple single compound marker model, which was very successful, but it's the low-hanging fruit. Similar happens in chemical ecology, where you had single pheromone systems that look really nice. And the best examples you find in fishes, you run out of pheromones quickly, it's all sort of steroids. The ones that really give the specific, species-specific cues are completely biologically non-active, individually non-active compounds, which provide the context. And I think context is also something which helps a little bit getting to grips with the environment. Because after all, organisms are famous for detecting differences and not absolutes.
Fantastic, wonderful. Any right at the back?
Thanks so much. I'd first of all like to say, Billy, that I don't want to just have one chip. I think that there should be 2. I think if you work from both ends, you stand a better chance. Yeah. Secondly, I'm not medical, but I get the impression that at the moment it seems very much a one-to-one relationship that's being drawn here between there's one biomarker for each thing. Are we not running the risk of not identifying interaction between them? And also, are we not missing the opportunity of seeing dynamic change in time, also producing useful information about disease and particularly early warning and pre-diagnosis? If we don't start looking at the patterns and looking at the ways in which these highly complex mixtures and changing mixtures are acting within diseases.
Excellent. Any other perspectives on that question?
Billy?
Yep. Paul, while the microphone's coming up.
Okay, so here's a nightmare that I have, in that you have a beautiful panel of markers released from deranged tissue. It goes into the bloodstream, is absorbed into the gut biota mess around with it. They then put it back into the bloodstream, it goes to the liver, and then finally it goes into the lung. Parts of it partition through the lung, and eventually you get to see 1/10 of 1% of what was the original signal, massively deranged. So you're absolutely right. And there's probably markers of this person is not very ill and is feeling miserable versus there's this very specific derangement in tissue that's producing a strong signal. So you're absolutely right. But the huge complexity of what we're attempting to do sometimes makes me feel very small and inadequate to the task.
Yeah, just one comment I was going to make. Coming out of the metabolomics background where I spent 8 years of my career, is I'm not seeing a lot of systems biology types of approaches going on within the VOC field. And I think, given that we don't really understand a lot of the biology behind how these VOCs are being created, I think there could be a lot of merit in terms of combining VOC research with metabolomic profiles and potentially other types of omic profiles, so we can look at the correlations and start trying to understand where the biology actually is originating from. So I'm very much in favor of understanding the biology, perhaps not just using VOCs, but combining it with other types of analysis as well.
Yeah, and I just to add to that, I felt in the discussion sometimes 2 things seem to be a bit convoluted. There is the untargeted discovery, where you try and measure as many molecules as you can and take a systems biology approach. I definitely agree that has value, and to some of the other speakers' point as well, taking that as a basis to try and understand the biology and is a very powerful strategy, because it allows you to find interesting candidates that you may not consider before, such as limonene, for instance. However, I would contrast that with saying, if you do a purely pattern recognition-based approach with what's typically called an eNOS type sensor, and if you completely ignore all biology and try and push just an eNOS-based approach to the clinic without ever trying to understand the underpinning biology or being able to calibrate it, that's something I think is fundamentally a waste of time.
Hang on, we need a microphone. Microphone was going to hear first. Ollie, here first.
Sorry. on. So I just wanted to return. I think one thing is really, apart from what makes genomics and proteomics so successful, is actually that there are publicly available databases of gene sequences. And so what Paul suggested, having profiles and finding a way to code profiles in a sensible way. And by profile, I mean the chromatogram, because there is a time dimension of all these compounds. I mean, it's sorely neglected in a way, and it's a vital piece of information if you have that as well. And then you can, it might not be the perfect coding, but if you can draw on other people's profiles, it would be such a nice thing. With the coding metadata, you can model them into your shape for your instrument. So that's getting increasingly more possible. But I think that is something which would be really, 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 really helpful. Never mind that you have to pay for the mass spectral libraries all the time, which is also not really. And that's a big difference, is you just blast a gene sequence. No doubt about it.
Yeah, thanks. I think it's pretty aligned with what has been said. Patterns, I wasn't referring to ENOs or specific system, but it could be a combination of individual VOCs that we identify, but they work together as a group for better statistical power also. So it's still based on understanding these compounds, trying to relate them to biology and biochemistry, not just quantitative patterns or qualitative. Sorry.
And even if you are able to measure a compound reliably and validate it and don't know where it comes from, ultimately that could be useful in clinic, that's fine. But you need to know what it is.
Dalia, then Bruce.
Thank you. You might think I'm biased. I work with Paul. I've been trained the 2 rules of assume nothing, trust no one for the last 4 years. We actually have those 2 rules on the board in every single group meeting. So, but I just wonder, if we don't know what we're measuring, if I were to talk about ENOs, if we don't know what we're measuring and we can't calibrate it, when do we know when it goes wrong and it stops kind of detecting, or diagnose a disease accurately? And then at that point, it's already in clinical practice, it's already at, point of care. We may end up misleading diagnosis. I don't know. Whenever I think about it, I'm a big supporter of understanding biology, understanding metabolic pathways, know exactly where that compound is coming from. Mm-hmm. So I think I just wanted to highlight that. Yep. We wouldn't know when and where.
Just a small comment on the discussion over there. I mean, in gene expression analysis, you have lots of, do a differential gene expression between 2 states. You have a ton of genes which are different. The way to reduce the noise and get some idea of function is to do either a pathway analysis or a master regulator analysis, looking at the regulons that may be in common between these genes. And presumably, as I think was being implied there, one could apply the same approach to looking at the molecules that come out of the VOC analysis.
Okay, so we're going to open it up for any questions that you might have for the panel and others in the audience as well. Any burning questions? Otherwise, I'll have to ask another one. Yep.
I have a question and a comment at the same time. I think that the word biomarker is largely abused. And I agree with Jonathan that we have no biomarker, for example, for lung cancer. But if you look at literature, you find tens of papers in which biomarkers of lung cancer have been shown and demonstrated and validated and presented. And this is almost valid for anything. So my question is, how do we fill the gap between what is actually known in the community and most experts in the field who knows that most of the literature is really flawed?
Yep. Good question. Who wants to take that one first? passing the hot potato.
Just because I was mentioned, right? Just because I was...
You're the editor, right?
I'm not responsible for all the papers that get published there. Yeah, so, I mean, you mentioned what I mentioned. I think, and actually coming back to the comment of the gentleman at the back, I think in the community, no one now expects that we're going to find a single biomarker that's going to represent one illness. So if we find anything, it's more likely going to be a combination of different chemicals, and probably they're not going to be uniquely present, but just present in different concentrations to the norm. So I think that's something that is of growing interest in the community, looking at perturbations, personal monitoring. When do things kind of stick out as being different to your normal breath profile, and does that then raise an alarm to specific diseases? The other thing, and that was something that Anil mentioned yesterday, is that when we're doing clinical studies looking at specific diseases, let's say lung cancer, versus a control, we're always taking healthy controls, but you would also want to have to look at perhaps people with other forms of cancer as a comparison, and not just completely healthy people. So I think this is something to look at. I don't know if that's really answered your question.
Does that address the question of it's practically impossible to reproduce results at the moment? A lot of things that go in, they don't agree with each other, and nothing gets reproduced, and nothing's getting validated. So I don't know if that would address that problem.
I think that it is a matter of fact that it is very difficult to reproduce results between different groups, and I believe also within the same group, because there is a large variability of the samples, the biological variability. And I'm not expecting, just to make unhappy our colleague from Madrid, of artificial intelligence to play a major role in the coming future, because the fact is that we do not have enough data to train the algorithms. The amount of data, if you look at most of the papers, you have tens of patients, and there is a limitation because of the money that each group is capable to invest for a study, and the number of patients that it is capable to enroll. And it is quite complicated to exchange samples and run multi-center studies. Yeah. And this is a large limitation, according to me.
Okay.
So maybe I can just address that, because I did. Apologies for drifting away from answering the actual question you said. So I think the International Association of Breath Research, we have a focus group on standardization. This is something that's been lacking in the field. Because of the challenges of standardization, you've got multiple different tools, multiple different diseases you're looking at, and multiple different potential biomarkers. And that's something we're addressing, and several years ago, Paul set up this study to have a kind of a benchmark for comparing different techniques, and that's the peppermint study that's now ongoing. I think we have 15 to 20 different groups that are involved in that, and similar to the EVOC talk we heard from Isabel yesterday, administering an encapsulated peppermint oil and looking at the release kinetics over time, and comparing one technique to the next in terms of analytical techniques, but also sampling techniques. And I think that's a great step forward to benchmarking and to see how do our methods compare, not just from sampling, but also for the analytics. Paul, I don't know if you want to.
Well, how do you disentangle the biological variation, though? Because you effectively you're testing different metabolism.
Well, I'll answer that question first. It's very difficult, and you don't want to. You want to show the biological variability. That's very helpful to the community. And certainly, I think the person best placed to answer this, although I won't ask them directly, is my colleague Dorota, who's been doing exactly that. But when you have 15 groups all contributing 10 times 6, so 60 times 15 groups of data, you're starting to get a general human model, and you're able to split out the different phenotypes within that group, and you start to see the variability, and that's also helpful. And you are able, despite that variability, to come up with a minimum a minimum benchmark that says if you can't get to this point here, then you're doing something wrong. So if you can't detect by this technique the following compounds in breath 6 hours post-ingestion in all of your subjects, then there's something the matter with what you're doing. And that ratchets up the standard of that group. If you find that you'll be able to detect these 6 compounds way beyond the maximum boundary that's reported in the original paper, you're doing something that is a significant advance on the art, and you should perhaps share that or patent it or do something because everyone would benefit from that knowledge. So it has that ratcheting effect. But I wouldn't try and hide the variability. I would say it's there to be understood, and this is one really good way of understanding it.
Yeah.
And I'll shut up.
Yeah. Isabel?
Yeah, I have more of a comment referring back also what to Paul said. Basically, if we don't know what our normal is, if we don't know if our healthy controls are comparable, how should we expect that our lung cancer markers are the same that we find? So we really need to understand what is normal breath in order to understand when do I see which variation. And then there's another, it's more of a comment to this bottom-up or top-down thing. I come from a systems biology background and did research in quite different systems, actually plants and bacteria, but we started to realize that very often when something disturbing happens, basically you have a homeostasis, and so the organism is functioning perfectly, and then there is an event or a disease or something, and the whole thing gets out of rhythm. And there's a lot of very small changes that are not significant on their own, but together they make a new phenotype. So that would be something where pattern recognition really could work, and where looking at individuals would not help you at all.
Giuseppe?
So just to follow up to Billy's question, so we may overcome biological variability by looking at more pathways, so we need more probes to diagnose the same disease. Because in this way, so it's very unlikely that for several pathways, so the variability is going to go in the same direction for all the people. So I think this is one way to go. And of course, I would like to know your opinion about my idea.
Mm-hmm. Any comments on that, Mark? You want to start?
Completely agree. Yeah, I think that fundamentally increases your resilience, and I think it deals upon an important point which was raised in the audience and also in the presentation today. And I can say this because I'm used to be a doctor, at least. The simplistic stickers that doctors put on patients calling it asthma is not a reflection of biological reality. So we should never expect that we can, with a targeted molecular mechanism, really capture all the variability in these syndromatic diseases, other than if it would be a genetic disease with a single mutation that is very well understood.
Any last comments on that before we wrap it up?
Maybe just to like to make a comment back to your comment on lung cancer, and I'm not at all an expert in the field, but you mentioned that there is a huge variability and not reproducible results. I think it's also important to realize that one biomarker has to be linked with a clinical question. And as mentioned by one of colleagues there, I think the question of biomarker then can predict occurrence of cancer in smoking people may be completely different from the question of diagnosing lung cancer, and also from predicting a relapse of cancer once you get operated. So I think we have to be very clear in which clinical question we ask when you think of biomarker in lung cancer. I think it's maybe a reason why there is so noise and lack of clarity, maybe. I don't know the literature, but I think it's clearly different points.
Okay, very last point, Paul, and then we have to wrap it up.
I want to reflect on a conversation I had at lunchtime. I want you to imagine a beautiful polished silver platter out in your garden. It's a beautiful April day and the rain is falling gently onto your silver platter. And as each drop hits the silver platter, it explodes and forms lots of little micro drops on the silver plate. And they're all separate and isolated. It keeps raining, and eventually you start to see the drops coalescing. And it's a chaotic process. They sort of expand rapidly, and 4 or 5 drops that were close to each other and separate suddenly become joined. And before you know it, you have a pool. So the metaphor is obvious, or the analogy is obvious. Right now we're waiting for these separate, isolated drops of knowledge to start to coalesce to form this pool of wisdom. And I think that's what we're about, what we're all seeking to achieve. I just come back to Jan Frederick here. Although I don't trust anyone, I love you all.
Excellent. Thank you very much, all.